This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 50, recorded on February 14th, 2013. Hello, everyone. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
It's Valentine's Day.
It is.
Here in the U.S., right?
Absolutely.
My daughter told me it was in honor of St. Valentine's Massacre. Is that right? I said, why is a loving day based on a massacre? She couldn't explain it.
You're right. I think your daughter needs to reread that that myth.
How have you been, Dickson?
I've been very well. Thank you, Vince.
This is an unprecedented 2 weeks after the last, or 3 weeks after the last TWiP. This is number 50. Congratulations. I'll shake your hand.
And to you.
Should we stop here?
Sure.
Should we give him the last episode at least?
Let's give him the last episode.
Okay.
It's ironic in some ways to be discussing some of these topics today, as we will see, because one of these organisms has a profound effect on the heart.
Oh, that has to do with
Yeah.
Valentine's Day, you
Yes,
mean?
exactly. So if you want an enlarged heart and show her that you love her, catch Trypanosoma cruzi. No, that's actually not what I'm recommending at all.
I have to get one of these papers up here. But I'm using a different piece of equipment today.
You are. I noticed that you're using your iPad.
You know why? Because
Tell me why, Vince.
I'm facing you. Would you like me to go?
No. This is
away?
much more amenable to a conversational.
I don't have my laptop, so I haven't.
Yeah, well,
An iPad, but
that's a half a laptop.
it's slower to get the papers up,
That's okay.
and you can't have a lot of things up at once.
No, you can't.
TWiP.
You can't do multimedia all.
Oh, you know the problem here is that
What?
this thing doesn't have internet.
Oh no.
The papers are here,
Good.
and we do have email today as well.
Excellent.
All
Looking
right,
forward to it. So we're going to do.
2 papers, right?
Yeah, we're going to do 2 papers.
Now, I picked one and you picked the other.
I did.
Is this how we're going to go forward?
I would like that.
Why don't we get some guests too?
You know, here's what I would suggest too, out in our listening audience.
So we're not stopping. We're actually going to
No, we're
continue.
going to continue. Of course, we're going to continue. Please don't ever say that. We're going to continue. Everybody on point here.
Unless you die.
Well, that's another issue.
And I
of
promise
course.
you, if you pass,
Yes.
I will not
You mean
get
when?
I will not get a substitute. I will
No,
end the show.
no. There's plenty of people out there that would be glad to do this.
No.
I'm sure of
I
it.
could never work with anyone on this show. It would have to be a different show.
This is a Valentine's Day show, isn't it? It was just a big love fest.
What's your idea? going
My
forward?
idea is that we could solicit from the audience topics and papers
Well,
that
we only
they
have 10
find.
listeners. It's not going to work.
That's true. I never thought of that, Vince. That's true. But maybe we can take suggestions from the audience so that next time we can feature their pick of the week in terms
they
of
actually
papers.
do make suggestions. We just ignore them usually. We've had suggestions for discussion papers in the past.
We have.
Maybe we even have one today.
Maybe.
So I could go back and pick some of the things
You
that they've
could.
told us to discuss. But anyway, so you want to make this an official call for things to discuss on
I would
future
love
TWiPs?
that. I would love that. Not that we're running out of ideas, mind you, but, you know, it gives us an idea about what's important to the listeners as to what they would like to hear about, too.
You would like to engage our listeners.
I would like to engage our listeners.
All
That's
right.
exactly
We
right.
can
And
do
since
that.
we don't take telephone calls and we do emails, which are delayed by weeks or days after the podcasts.
You know what one of our problems is? We don't have a regular
we have
schedule.
many problems.
I would
Vince.
like to get some guests. I have some ideas for
Well,
guests to get on the show. In all different areas of parasitism, because it's a broad field, as you know.
Of course I do.
It's
that.
not just about trypanosomes and
No,
trichinella.
and malaria.
I like those parasites, the nematodes that go into the foot of the clams.
All right.
And I know someone who could come and talk about that with us.
Exactly right.
And in fact, when I get off the horn here,
Yeah.
I'm going to send that person an email.
Excellent.
But I don't know when you're available next. That's the problem, Dickson.
Well, Vince.
See, on TWiP, I know every Friday at 2 p.m.
That's true.
we record, and I can tell people, can you make it this Friday or that? But with Dickson, I just don't know.
But you know what we might do? My wife suggested something the other day that might be applicable here, and that is that we might want to record several in a row, like on the same day, and then release them one at a time, 1 week apart. So they do that a lot for series on television, for instance. They film them all at once, and then they release them one a week. So we could do something like that when my schedule doesn't permit me to be here, that's all.
All right.
It's just a suggestion. I don't know if anybody out there is listening or whether they care, but maybe you should send us your suggestions for that, too, because we would like to be on more often. But you're right.
Mean, I have never recorded multiple shows in a row because after one, frankly, I'm exhausted.
You're burned out.
I'm exhausted.
That's right. You've had enough.
I used to do TWiP and TWiM consecutively on Fridays, and it was too tiring.
Too much. It's too
Because
much.
I'm 60. I'm an old man.
Are you really? I didn't know you were that old. You look 59 at most.
Did I tell you someone
It's a bad
came
joke.
up to
It's a bad
me
joke.
at study section,
Yeah.
a young virologist,
Yeah.
and he came up at dinner, and he shook my hand and said, I just so much want to meet you. I read your papers when I was a student,
Yeah.
and he said, I thought you would be 80.
You said, that is my hope. That's what I was
So,
saying.
I posted that on our Facebook
That's
page,
funny.
facebook.com/thisweekinvirology.
That's funny.
And people were amused, and one person said, "What's wrong with 80?" It's true.
Yeah.
Nothing's wrong, but I'm not there yet. That's all.
True.
All right.
Don't rush it. All right, let's do the malaria paper first, I think.
Malaria paper was published in Cell, Host, and Microbe.
Right.
And I thought this had a broad appeal.
You were right, by the way, because...
We can go into a number of things. And in fact, Dickson, I have here my textbook, which is marked to the page on complement.
Excellent.
We have a nice figure, which...
Is that like when you admire something and then you give them a compliment?
exactly.
Ah.
Do you know why it was called... Well, we're jumping the gun.
We are.
The
aren't
title of this paper,
we? Yes.
Malaria Parasites Co-opt Human Factor H to Prevent Complement-Mediated Lysis in the Mosquito Midgut.
Incredible. Incredible.
Co-opt, malaria parasites, all these words. Factor
Yeah.
H.
Factor H.
This... Oh, I won't go there.
No, please don't. No, because
You know what I was
I
thinking.
know where you were heading.
Yeah, it's terrible.
That's preparation H, Vince. That's not factor H.
Complement is a first-line defense.
It is.
Against infection with
First
many...
line? First line?
You don't think it's first line?
No, I think the skin is the first line.
Okay. Yeah, I suppose.
Then mucous membranes second.
Well, if you're going to count lines, then we're going to be pretty far in with complement. So the skin, the mucous membranes, these are all considered non-specific defenses.
Barrier defense.
Barrier defenses.
That's correct.
Then the immune defenses, we have the first line,
Right.
and then the second line. We have the
Right.
innate and the adaptive
Correct.
defenses. And
Correct.
complement is actually part of both.
Yeah.
So there are some kinds of complement that act very early on and others later. So what is
Weird.
complement? Do you know? Did you learn complement in school?
I did. When I'm so old that they only had two factors then. No, that's not true.
It was discovered in the 1800s, I believe.
Yeah, it's an old system.
Let me see if I have 1890. The complement system was identified in 1890 as a heat labile serum component that lysed bacteria in the presence of antibody. In other words, it complemented
Yes.
antibody.
Yes.
What does heat labile tell you?
I guess it's sensitive to being raised above the body temperature level.
What does it mean
It means
biologically?
it's destroyed.
What would be destroyed by heat?
Proteins.
Yeah, probably proteins, right?
Probably.
What else is there?
Well, there's lipids and carbohydrates.
Usually in the old days, a heat lability was interpreted to mean
Yeah,
protein.
that's true.
Anyway, it is a very complicated system.
Yes, it is. It's hard to imagine this evolving, and yet it did.
Actually, so many things are hard to imagine. As I say often, you couldn't make this stuff up.
No, you couldn't.
It's so amazing.
Although some did while they taught because they didn't know all of it.
So there are two complement pathways. So complement is a series of serum proteins
Right.
that participate in immunity.
Correct.
And there are two pathways of complement,
Yes.
right? There's the classical, because you go to a theater to hear it,
Exactly.
symphonies.
That's
Classical complement and alternative. It's just like music, right?
It's just
Classical
like
music and
or,
alternative.
Or carbohydrates. Right.
What do you mean?
Well, degradation of carbohydrates. There's an alternate pathway for
There
that,
I
too.
bow to your expertise.
Oh, please don't do that. You'll break your back.
Now, in the classical pathway, antibody is involved.
Right.
So if you're infected with a pathogen, you make antibodies, which will take up to 2 weeks, right?
Yeah.
10 to 21 days, let's say.
Right.
And then the antibodies will recognize...
Wait.
Yes,
Is it dependent on which kind of antibodies?
probably.
Probably. So in the beginning of an antibody response, what kind of antibodies do we make?
IgM.
And is that a complement-binding antibody?
No, I don't believe so.
It isn't. Exactly.
IgMs are, right? IgG, sorry.
That's right. So it takes a while
Yeah.
for the IgM response to fade after it's elicited.
Yeah.
And then the IgGs come up afterwards.
Well, you did learn this.
I had Elvin Cabot for a
Yeah,
teacher.
right.
So I remembered everything he said.
These antibodies recognize
Yes.
foreign components
Correct.
like bacteria.
Right.
Let's say they recognize the surface of a bacterium.
Right.
Then there are other components of the complement pathway, lots of serum proteins. They would then notice that this antibody is bound to the bacterium,
Yes.
attract other components, poke
Yes.
holes in the bacterial membrane.
That's
they make
right.
these membrane attack complexes that
And they
poke
bind to
holes
the
in.
FC receptor on the antibody molecule.
Yeah, that's right. And there's a cascade that happens. It's not just one thing and boom, there's a protein binding, something else gets cleaved, another thing binds, boom, boom.
It's
We don't have
very
to go into it.
But it's like
that's why
enzymatic reactions.
That's why it's a
isn't it?
cascade, right?
Correct. It's called a cascade. Like the clotting cascade.
And I have to just say one member of this cascade,
Yeah.
one of the proteins, or one of the earliest proteins, is called C1q.
Right.
It's a protein that will recognize the surface. Actually, sometimes C1q can recognize the surface of a pathogen based on certain sugars or some, or it
Okay.
can recognize an antibody bound to the surface, which would be that would be an immune reaction.
Right.
There's a bunch of proteins called C1q in the collectins, which I always thought was a great name for a rock band.
Collectins.
Isn't that?
The
C1q
collectins.
in the collectins, right?
I heard them once.
From here every night.
That's right.
So now let me just say the antibody has to come up. We just said IgG is going to take a while, right?
It is. Well, it actually starts. No, IgG takes a while. I'm sorry. Yes, exactly right.
There is a complement reaction which can happen immediately because in all of us, so we have these bacteria in our gut that have sugar moieties on them to which we make antibodies. And these antibodies are a good fraction of our serum. Many pathogens that come in have the same sugar moieties on them.
Is that like LPS or something?
It's not LPS. It's a specific kind of sugar.
Oh, okay.
Um,
Related to pneumococcal polysaccharides?
It's called natural antibody. It is galactose alpha 1,3 galactose.
Ah.
Okay, it's a terminal sugar on glycosylated proteins.
Okay.
And when a virus or a bacterium comes in, you've already got those antibodies made in you already because they're in your gut.
I see what you mean.
So the antibodies bind, and then complement comes in, and boom. So in that sense, complement can be an innate part of the immune system.
Got it.
Right?
Yep.
I'm sorry I'm going on here.
No, go, please, on.
Do you have any questions?
Only later when you finish.
All right. Alternative pathway.
Right, that's an interesting one.
You don't need antibody.
Huh.
So this can recognize a microbe as foreign immediately and make a hole in the membrane.
And they all make holes, right, no matter which way they work.
So they make holes, but they also can attract. They can make proteins that attract and induce inflammation.
Right.
Not attract inflammation, that induce inflammation.
I know what you mean.
Okay.
Yes.
They can attract cells that then produce cytokines. So
Sure.
two things: lysing cells and mediators of inflammation.
Got it. And there are little pieces that are created as a result of these cascades that
they're
also
proteases
have...
that break pieces off and have activity, so they have to be activated.
That's
so I guess
amazing.
the cascade helps you to protect against yourself.
I was going to ask about that, Vince. How do you protect yourself against this innate complement fixation reaction?
Well, there's some... we have proteins in us that are complement inhibitors, all right?
Right.
Because you don't want to lyse yourself, right?
I think that's a great idea not to do that.
Right. In fact, let's see. Oh, no.
What? You pushed the wrong button.
I want to tell you about the control of complement.
Right. And by the way,
where is it?
another way to know that it's not an acquired immune response is to know that the same level of complement is present before, during, and after the infection. Is that correct?
Yeah, that's
so
true.
So it's not inducible.
It's not induced, but the components they recognize foreign things and they're cleaved and new things are produced, but nothing is induced as far as I know.
So when they're being used up, is there not a feedback to make more during that time? So
I don't know the answer to that. That's a good question.
Right.
So we basically have a lot of we have factors that prevent self-damage. Okay.
Right.
We have regulators of complement, so we don't complement ourselves.
Exactly.
So to speak.
Exactly.
Right. And one of them is called FH.
You remember what Emerson said about self-praise, don't you?
What did he say?
He said self-praise is no praise.
Yeah,
so you shouldn't compliment yourself. That's what that means.
I try not to. I find it not.
It's not
good.
It's not Vince. Vince just doesn't do things like that. Nor do I.
So this one complement control protein called FH.
Right.
It regulates these alternative pathways. So the alternative pathway is the problematic one because it doesn't depend on antibodies, right?
Right.
So you could have complement binding to your own cells and lysing them. So
That would be
we have
horrible.
regulators that prevent that. We basically antagonize some of the early complement proteins so that they don't antagonize us.
Right. And yet they're still active for whether they're needed.
Apparently they are still active when they are needed.
Right. So we don't destroy them. We just prevent them from binding to our own cells.
So I think the idea is when a pathogen comes in, then a signal is sent.
All games,
let's go.
The games change. That's right.
But I think you still protect yourself, although maybe you have some cell damage in the infected areas, which is part of immunopathology.
Exactly right. That's called innocent bystander response, yes?
Now, a number of pathogens have evolved to interact with these complement regulatory proteins.
But of course,
right?
Absolutely. There's got to be a yang for every yin.
Absolutely. So the immune system is so good that if the microbes didn't have antagonists, they wouldn't exist.
Exactly.
Right, because the immune system is just great. So every virus genome encodes at least one antagonist of the immune response. So many microbes they bind these complement regulators, and they mimic the host cell surface so that they're not. Isn't that incredible? Isn't that great?
That's amazing. That is amazing.
So they're protected. So a lot of bacteria have complement regulators. So basically, this paper. A lot of bacteria have proteins that bind complement regulators.
Understood.
This paper examines malaria because, as they say, complement evasion mechanisms are not well described for parasites in general, and they're not known at all for plasmodium, which, of course, is the agent of
malaria.
Malaria is how many people a year? 200 million?
Oh, it's much more than that, Vince.
I read a book which says that in certain parts of Africa, every kid at any time of his life has some malaria parasites in his or her blood.
I would believe that. I mean, Nigeria alone has 70 million people, and that country gets malaria every year, basically. So according to a good friend of mine, Jim Jensen, the number is more like 2 billion. 2 billion people, not just at risk, 2 billion people infected.
Now, they make an interesting point here.
Go on.
The compartmentalization of plasmodium in erythrocytes is a means of evasion.
There you go.
Because if you're in an erythrocyte, you can't be attacked.
Exactly right.
But at some point, you have to get out, right?
You do.
Can you review? You get bitten by a mosquito.
Sure.
The schizonts come in? Sorry. I love that word.
They don't quite come in yet, Vince. The first thing that happens is that the sporozoites are injected into the blood.
Right.
And they travel around seeking out the
Susan. They seek out Susan desperately.
Desperately seeking Susan. And when they find her, namely the sinusoidal cells of the liver.
Which happens quickly.
Very quickly.
Because all the blood goes through the liver very quickly, right?
Yes.
So you get bitten by a mosquito. If it injected a dye, that dye would be in your liver within seconds, right?
Yes.
Amazing.
It is.
So it gets in the liver, it seeks out a sinusoid.
Yes, and there's a specific protein receptor ligand interaction.
It burrows into your liver?
It doesn't do that yet. It binds to the sinusoidal endothelial cells.
Okay.
And then it's transported across that barrier to the cuboidal, the hexagonal parenchymal cells of the liver.
Okay.
And they penetrate a single cell.
So now they're evading the immune response because they're inside
It's too soon. I mean, it's like minutes.
Yeah, there's nothing that can
There's
happen
no
anyway.
way that an acquired immune response could act against them. So, and they get in, and then they start to change. and they develop into what's known as a cryptozoite. Crypto meaning you can't see it. Okay, because if you look in the blood, it isn't there. If you look in the blood, it's not there. If you look in the blood, for how long? 9 days for some of these malaria parasites, and even longer, 12 or 14 days for other species. It takes that long for the organism in the liver to make enough cryptozoites to fill up that one liver cell and burst open and release what are now called merozoites.
Where are the schizons?
I'm getting to that, Vince. He loves it. Your favorite part is almost here.
So they burst open, merozoites
come out, but now they have lost the ability to reinvade liver cells.
Okay.
Thank goodness, because if they could do that, it would be a disease of the liver.
It's not.
But it's not a disease of the liver.
You don't get yellow?
You do not get jaundice.
Well, that's yellow fever.
That's true. It's hepatitis also, by the way. So where do the merozoites go?
I would assume they go back into the blood.
They do, and then what happens, Vince?
They mate?
No.
I'm guessing. I'm sorry.
Each one attaches to a red cell.
Oh, they go into the blood and they find red cells.
Right.
They attach and penetrate the red cell.
That's correct. And some of them have specific molecules that they use to recognize the red cell with, like Duffy blood groups and stuff like this. So in goes the parasite. It's actually ingested by the red cell. Remember we talked about the parasitophorus?
Yeah, I do remember.
This is a cell without a nucleus, and it's making a parasitophorus vacuole.
Only erythrocytes, right?
Yeah. It's amazing.
Without nuclei.
Correct. Now, except if you're a bird, malaria. And
I was
then,
going to
of
ask,
course,
chickens have nuclei in their
they
red
sure
blood
do,
cells.
and so do ducks and canaries. And remember, Sir Ronald Ross worked
So
on bird malaria.
In that case, do they still go into the nucleated red blood cells?
They do.
Okay.
They just push it aside. So here's this parasite inside the red cell.
All right.
Now comes your schizons. His eyes just lit up. Oh, good, I get to talk about my schizons.
Well, I'm not going to talk. You're going to talk.
So schizogony, or schizogony, depending on how you like to pronounce it, is the process by which the organism first grows a very large amount of cytoplasm. Mm-hmm. And then the nuclei inside that same common cytoplasm divides, and then a miracle happens. Or at least we don't understand what happens. The cytoplasm starts to partition itself off and sequesters a nucleus in each little piece of sequestered cytoplasm. And at the same time, Vincent, remember what it produces as a byproduct. It uses hemoglobin as a food, and it produces hemozone as their byproduct.
We talked about that last time.
We did. So here's this single cell with multinucleates.
Yeah.
Nuclei, and in the middle of it is this toxic waste dump that it's created by eating your hemoglobin.
Right.
As it pinches off the cytoplasm, it excludes the hemozone. Mm-hmm. Clever little organisms.
Right.
And then they burst out of that red cell, and then they can now attack another red cell.
Is this the sexual phase?
No.
This is
they're haploid at this point, as Rich Condon says. I knew him when he was haploid. So now they're haploid, and they're haploid until they get into the mosquito. All right, now, that doesn't involve schizogony. In fact, some of these merozoites, when they go into a red cell, they don't divide.
Okay.
What do? They begin to differentiate into pre-sex cells called gametocytes. You've got a micro, which is the male, and the macro, which is the female. The micro... Now all of this doesn't take place in the blood of the victim like us. Now the sexual stage has to come into play. Now, where does the sexual stage come into play?
Mosquito.
Correct. So who's the definitive host for the parasite?
The mosquito.
Yes. Isn't that ironic? We are just a carrier for this parasite. The mosquito is the big victim.
And they're fine, though. They don't get sick, do they?
Didn't say that. I mean, if they ingest enough blood with enough gametocytes in them, some mosquitoes can actually get overwhelmed by the infection and die from it. Okay? But that's an enormously heavy infection, by the way. So what happens next is that the mosquito's stomach contains digestive enzymes.
Okay.
But it also is filled with human blood.
Yeah, of course. Which has what in it?
It's got complement in it.
Exactly.
So what happens now when the mosquito digests away the erythrocytes, revealing the parasites?
So it could be susceptible to complement, right?
It could. And in fact, this paper does a very good job of exploring that capability. Because
how long will the complement last in the mosquito gut? It's the gut, right?
It's about an hour. They said it's about an hour. I read this paper too, and albeit quickly, but nonetheless got the gist of what's going on here. It takes about an hour for the blood meal to get completely digested.
Right.
But in the meantime, as the microgametocytes are released, and the macrogametocytes. The microgametocytes actually undergo a process of exflagellation. Now, Vince, let me ask you something here.
I'm listening.
You know the Nobel Prizes, right? We know we know two people here who have Nobel Prizes. Actually, you know three people at Columbia University that are not personal friends. One of them is Marty Chalfie, and the other, Richard Axel, I know pretty well, but Racaniello is the third person. Now, if they were living at the turn of the century and they were interested in working on malaria, and they had discovered that when you take blood from an infected individual and you just leave it, let it go into anaerobiosis, what happens at that moment is that some of these parasites in some of those cells develop what looks like long filamentous structures. And under the microscopes of those days, it looked like a little wiggling sperm. In fact, it is a sperm,
Right.
because these are haploid. So
Right.
these little sperm now try to inseminate, basically, the macrogametocyte, which is the female equivalent. The next thing you know, you've got a sex cell. You've got a zygote. But they don't call it a zygote. Naturally, why would they call it a zygote? They call it an ookinete. But I want to back up. The Nobel Prize for discovering exflagellation, right, was given to Laveran for discovering that. That was
Laveran,
Laveran got a Nobel Prize.
Yeah, sure.
It was fantastic, and that's what he got it for. So in the mosquito gut, the mating of this organism occurs.
Right.
But there are these proteins there from the host that says,
From your blood meal, right?
Yeah, oh no, you don't. But what else is there too then?
Well, the malaria parasites are there.
Factor H is also there.
Factor H would be there.
Has to be.
So the question here is whether the complement acquired by the mosquito is going to harm the malaria
Is it
parasite.
quicker than the ability to acquire factor H?
We already know the answer because this is a successful parasite.
They
right?
did the work and proved that the parasite, when it's released from its host cell and undergoes morphogenesis to become a sex cell, also picks up factor H on its surface.
That's right. It binds the surface
And
of the So what stage is now binding factor H?
This is the now male and female sex cell.
Okay.
You would call them that. Because when they get together, it's called the ookinete.
They call them macrogametes and young zygotes. They both have
Young
something
zygotes.
on the surface
That's
that
right.
binds factor H.
A young zygote.
Remember, factor H is this antagonist of complement.
Exactly.
So this is brilliant,
it's
right?
a cloaking device.
And it's another cloaking device. They want to protect themselves from complement in this 1 hour where the complement
It's amazing.
is active, right? It's pretty cool. It must have evolved to select for parasites that express this protein, which they actually
Remember
identify.
whose parasite this is. This is the mosquito's parasite, right? So
But it's human blood.
I know that, but
It's
before
not
there
mosquito
were...
complement. Why don't mosquitoes have complement?
I didn't say they didn't. There are probably complement-like molecules.
Maybe not in their gut.
No, it's in the hemocoel. It's in the circulating clear fluid that they
Right.
call blood. But
Right.
but okay, so
Now, I should say that this complement pathway that's active in the gut
Yes.
is the alternative,
right.
right? Because unless the mosquito ingests antibodies from someone against malaria, and they actually did an experiment where they showed that if they add blood, so they have an interesting assay here. Can
They
you
do have
explain it?
They have a fantastic assay.
They're measuring the number of.
Well, they use a mosquito. They have to use a real mosquito. So
So how do they do this? Explain.
They raise mosquitoes in the
Right.
laboratory. They're called Anopheles stevensii. And Anopheles stevensii's genome has been sequenced completely, right? Because it's looked at as the laboratory equivalent to Drosophila, but for malarial work. So the human genome has been sequenced, the parasite sequenced already, and now the mosquito has been sequenced. So we know a lot about this mosquito species, okay? So what they do is they take infected blood from a patient,
Right.
and then through a membrane, they allow the mosquitoes to feed.
Right.
And that's their assay in these cages of caged mosquitoes.
And then they're counting something in the mosquito, right?
Well, they count ookinetes.
And that's a measure of whether the complement is active or not, right?
Exactly. But let me just describe one more stage to this to fill in the listeners again, to remind them that once this mating has occurred, and that occurs extremely fast, like within the first 5 minutes, once the zygote has formed, it then penetrates out of the gut between the gut cells. It doesn't go through the gut cells. It goes between them
Right.
and comes to rest under the membrane that holds the entire gut tract together as a sheath. That's where this infection finishes its life cycle in the mosquito, because at that point it starts to differentiate and make sporozoids, which is what the mosquito is going to inject, right? It's only there for a couple of minutes. So it has to dodge bullets. It's like getting into a firefight.
Yeah.
And in a second, you have to cloak yourself, mate, and get out of the intestine. It's incredible. I mean, there's so much selection that goes on at each one of these levels that it's a miracle that it ever happened. But look at how popular it is, right?
Now explain this to me.
I'll try. I'm not sure
They
I can.
count the number of exflagellation centers, which they say is an indicator of activated male gametocytes,
Correct.
macrogametes, right?
They call them macrogametes. Okay. Maybe
So what's the number of exflagellation centers? What is that?
Well, when they make a smear from the
Mosquito?
the blood in the gut of the mosquito,
Yeah.
they get these little pools of blood and they look for an exflagellated.
What's exflagellated?
It's when they start to develop these sperm-like
okay.
critters that then break off and each one seeks out a female.
So they count those as a measure of whether complement is active or not, basically.
Right, because if there are no exflagellation sites, then they get killed.
So you know what they did was cool? They put an antibody to factor H.
Yes.
And what would you think if you add antibody to factor H?
You
what would
screwed
happen?
up their cloaking device and
Yeah,
you
so
kill
you
them.
get fewer exflagellating centers.
You can fire your phasers.
It's amazing. It's pretty cool.
It's
I mean,
wonderful.
that's a good antibody, obviously. to
It's
use for
exceptional biology. It's great biology that they were looking at. By the way, as an aside, there is a vaccine against the ookinete. They make antibodies against it. Forget about the factor H and complement. They just make antibodies against it and then allow mosquitoes to feed off of the blood that contains the antibodies, and that prevents transmission. So they have a vaccine already.
that. Yeah, in fact, in this paper,
Bob Gwadz and a good friend of mine worked on it.
they give mosquitoes a meal with antibodies to that.
Yeah.
And they show that the classical pathway is activated in that case, which is what
Exactly.
you would expect, right?
You would. And
remember
so
that-
factor H doesn't prevent that from
Right.
going forward.
It's very cool. So factor H binds the malaria parasite.
Right.
Or let's say at this point we know it's involved. We don't know if it binds because they haven't done the experiments. But
Right.
in the next series they do some immunofluorescence to show that if you have an antibody to factor H, it's on the surface of these sexual stage parasites.
Correct.
Right?
Correct.
And that's pretty cool.
It's very cool. By the way, I was always under the impression that the terminology changed in the gut of the mosquito the moment the parasite was released from its red cell. So if they call that still the micro or macrogamete, I would have changed that to immature sex cells.
Okay.
Right.
Now the protein that's binding is called C3b.
C3b. So that's before the alternate pathway.
So that's part of the alternate pathway.
That is the alternate pathway.
Yeah, so C3
Okay.
is cleaved spontaneously in plasma,
All
and
right.
then to C3b, which binds the surface of the malaria parasite.
Okay.
And C3b is inactivated by binding to factor H. Okay, so factor H is sitting on the surface of the malaria. C3b is coming. It wants to inactivate the parasite, but it sees factor H. Boom, it's inactivated.
Right.
Isn't that clever?
It's
And
enormously
they do
clever.
experiments to show that this happens.
It's wonderful.
It's very cool.
And they're using Plasmodium falciparum, which is, of course, one of the most dangerous species.
Is it falciparum or falciparum?
It depends on which country you're from.
Now, let me just look up one more thing before we go to the next cool thing.
If you're from England, it's falciparum. If you're from the United States, it's falciparum.
So factor H causes conversion of C3b into a cleavage product, and that's how it's inactivated, basically.
So you can use it up just by interacting with factor H.
I suppose, yeah.
If
I
you don't
suppose that
recruit
there's a
more,
yeah, because
you
in the
get a
blood meal,
finite-
there's a finite amount.
There is.
Now they identify the protein that it's binding to.
Interesting.
Do you know how they did that?
A Western blot.
Yeah, so that would be the first step. You could do a Western blot. They then added FH, factor
Yeah.
H to
Yeah, yeah,
malaria.
yeah,
They
yeah.
immunoprecipitated and looked at what protein was there. They actually used mass spectroscopy to sequence it, and they found out it's a protein called PFGAP50.
So they had a name, or you know why? Because they've already sequenced the genome.
Yeah. It is a glidosome-associated protein. Glidosome-associated protein.
I heard you.
And the PF is Plasmodium falciparum. GAP is glidosome-associated protein of 50 kilodaltons or 45 kilodaltons. What's a glidosome, do you know?
Well,
you could say I don't know.
No. I had a good friend who studied gliding in diatoms.
But this is, I hate to break the news, Dixon, but this is
It's not a diatom.
It's not a diatom.
It's got plant genes in it. Plasmodium falciparum.
The apicomplexa genes.
No. Plasmodium falciparum actually has some plant genes in it.
But isn't the apicomplexa a chloroplast?
No.
No?
No.
Mitochondria or bacteria, apicomplexa or chloroplast. No?
No.
You said that once
I
before.
never said that.
All right, so what's the apicomplexa?
No, it's a group of parasites.
No, there's a thing in the parasite called an apicomplexa.
No, it's not. That's the group is known as an apicomplexa.
I understand because they have an apicomplexa, don't they?
They do. No, but they have a complex in the apical end.
Right. That's what I meant.
Which are micronemes and rope trees.
Aren't they derived from chloroplasts? Rope trees. That's what Boothroyd was talking about.
Yeah, sure.
It's not derived from chloroplasts? No. What is derived from chloroplasts?
Well, plants. I don't know anything that comes from chloroplasts, but I think
in parasites.
There are some plant genes in Plasmodium falciparum. But the glidosome, gliding, let's just talk about gliding for a minute. What is gliding?
I have to go listen to the first 49 TWiPs again.
Pardon me. Me too, by the way.
Why?
Because I forgot what I said.
Okay.
But I know about gliding a little bit because a friend of mine used to study it in diatoms.
So this is an internal protein, right?
Well, it's on the surface, though, because the glidosome has to be inside, right?
Yeah, so but that means it must relocate, right?
What it's doing is it's allowing the organism, without any apparent visible means of locomotion, to locomote. Right? So it just glides along the surface, and it appears to be gliding as though it's skating on ice.
Right.
And so there's a collection of proteins that come together under the membrane, which provides the mechanism through which organisms glide. And I'll be willing to bet you that there's not a lot in common between the glidosome of Plasmodia and the glidosome, let's say, of diatoms, because those are two completely unrelated groups. But they may employ similar molecules because they all have to do the same thing. So you get convergent evolution. All right. So, and by the way, that's true for a lot of things. You'd assume that, pardon me, that the mosquito species that transmits malaria, that's the Anopheline mosquitoes, right?
Right.
They have a whole bunch of substances that they secrete into your skin when they bite you, which dilates the blood.
Pharmacopoeia, right?
Pharmacopoeia of materials, and a lot of them are very interesting. And the Anopheline mosquito group arose at a different point in evolutionary history to the Culocene mosquitoes.
Right.
And the Culocene mosquitoes have the same bioactive molecules, but they're all different. And yet, evolution says if you're going to be a mosquito, this is what you have to do. So I don't care how you do it, just do it. And so the Anopheline says, I'm not going to show you how to do this. You're crazy. I don't want the competition. So the Culocene says, that's okay. We have time on our side. We'll just develop our own system. And they did. So glidosomes may be the same in the terms of, I have to be able to move. I just can't sit here. I'm a sitting duck if I sit here. I have to move. I've got to be able to move. So glidosomes probably arose many times over evolutionary history to enable organisms without flagellae or amoeboid motions or cilia the ability to get around.
Now, what they say here is that normally this PFGA-P50 is located on the inner membrane complex.
Sure.
And then when the GCs are activated, germinal centers. No, not germinal. Tell me what is GC again?
Glidosomes.
No, not glidosome. GC is the stage that they're studying here.
The gametocytes.
Gametocytes, sorry. Thank you.
The gametocytes, I'm sorry.
When the gametocytes become activated, the protein goes to the surface.
How about that?
It relocates.
How about that?
So in other words, this has been evolutionarily selected for because the thing knows when it's in the mosquito gut and it's being activated, the GC, it has to put this on the surface to avoid
to bind
human complement.
Factor H, that's right.
So normally this protein, which again binds factor H. It's normally internal. It goes to the outside in the right place at the right time in the mosquito gut to bind factor H and avoid complement-mediated lysis.
I have a larger question to ask then that relates to this, and I would like to know the answer, and I'm presuming that now that this system has been described, you can go back and get the answer. You line up all the mosquitoes. You line up all the malarias. And you wonder why they're not all transmitted by everybody. What are the factors involved that gives specificity to the transmission cycles? Mm-hmm. And maybe we're looking at one of them. Factor H from humans permits this parasite to infect that mosquito species.
Correct.
But what if it was P. knowlesi? Plasmodium knowlesi comes from monkeys. And would this same mosquito be able to transmit Plasmodium knowlesi? I don't know the answer, but you can't line up all the Anopheles mosquitoes. They won't all transmit all the malarias. There are specific groups that transmit specific mosquito parasites. And maybe it's predicated on things like being able to attach factor H from a monkey or from a cervid or from some other animal.
I presume that
they can now do that study.
A monkey would have complement and it would also have factor H.
But not like ours.
It would be different, but it would probably, you would have to antagonize it in the mosquito gut, otherwise the infection wouldn't be transmitted.
Guess what? They can actually do that experiment now, too. They can take all of the primate factor H's, clone them, make the protein, and in vitro try to protect the organism from being lysed by the complement. It would be a fascinating thing to see because you could then start to look at the biochemical basis for host specificity. And remember, the mosquito is the definitive host, so we're really curious. We're more than curious about it. We have to know precisely why it is the definitive host. Why is the mosquito the definitive host in this case? Because it's unlike any other parasite that we know.
I can't see anything being done to the mosquito to take advantage of this knowledge, though.
You mean, why would the mosquito want this to happen?
Well, the mosquito is a bystander. It has nothing to do with... The only thing it provides is an environment for the activation of the GCs, right?
Yes.
But the protein in the GC
it does more
that
than
binds the factor H has nothing to do with the mosquito, right? That's a malarial evolution.
So,
it's an evolution to be resistant to human complement.
What you're asking, is there an evolutionary advantage to a mosquito to be able to transmit malaria?
Yeah, you're absolutely right.
I don't know the answer to that, Vince.
Probably not.
Who knows?
How many mosquitoes in the world are carrying malaria?
Well, in any given population, according to my friend Robert Quads, who we've had on our show in the past, and he's a big malaria mosquito expert, he said in any one endemic area, you've got less than 1% of the mosquitoes infected. But since you're infected 1,000 times during the day, each day, the chances are really good that you're going to catch malaria. So
1,000 times a year, some people.
Oh, no, per day.
Really? I thought it was a few times a day.
1,000. No. Some people get bitten 1,000
that's like
times a day.
Every minute. It's crazy.
They're always covered with a cloud of mosquitoes.
Wow. And we're covered with a cloud of viruses.
That's right. So the point is, could there be an advantage to that 1% of mosquitoes? And in fact, the malaria parasites actually shorten the lives of some of these mosquitoes.
They do an experiment here showing that if you put antibodies to FH in the blood and give the mosquitoes a blood meal, it inhibits transmission. It allows better transmission. Okay?
Say that again.
All right. So if you block FH, factor H, with an antibody, factor H is the inhibitor of complement.
It prevents...
You decrease transmission because the complement...
Oh, decreased transmission.
Did I say it increased?
You did.
Probably.
But I thought you meant decreased.
There's so many negatives.
You meant decreased. That's right. Let's be more positive.
In other words, if you antagonize the function of factor H...
Yeah, that's the gist of the paper.
It reduces parasite transmission.
Correct.
And they can do that by giving mosquitoes a blood meal.
Yes.
With or without the antibody to factor H.
Exactly. Clever.
So can we take advantage of that in some way?
Well, like I said, they've already got a vaccine.
I know how we can do this.
Go on.
We can immunize everyone against factor
H. Don't do that. Please.
And then the mosquitoes will take up antibodies, and they will have reduced transmission.
No
good?
I don't think so.
Because we will probably mess ourselves up by making antibodies to factor H, right?
But the thing is that maybe you could develop an antagonistic molecule, just the portion that binds to the surface of the parasite, a peptide.
I say that again?
Maybe we could synthesize a peptide which represents the binding site of the factor H to the surface of the mosquito.
PFG
Right.
AP50, yeah.
And then you could block the absorbance of factor H with just a little peptide that I still don't know how you'd use it.
They say that these GAP50s may be targets for transmission-blocking vaccines.
Well, you're running the risk of screwing yourself up at the same time.
Well, you're making an antibody to the plasmodium protein, but it's not going to prevent you from getting infected, right?
Oh, right. No, that's right.
But it's going to block transmission in mosquitoes.
They call this an altruistic vaccine.
Yeah, exactly.
Yeah, that's right.
It's not going to help you at all.
That's right. That's right. Exactly right.
Now, this is a little diversion, but I just want to get the clarification.
I'll try. I've confused you enough already.
Would an ideal vaccine in humans
prevent
the invasion of the liver by those initial schizons? No, schizons. What are they called?
Sporozoids. Yeah.
No way?
No, that's a good idea. In fact, that was the basis for the very first successful proof.
The Circumsporozoid vaccine was based on that form.
It
was.
The
6-his protein or whatever it was.
Only 4. It's a 4-repeat peptide.
And that was going to block the invasion of the liver.
Yeah, and in the data...
Dickson, you said earlier it happens so quickly.
In minutes.
How could you have an antibody response to block that? No way.
No, but you can't make one during the course of the infection. But people from endemic areas
they're going to have always
they have it. They have the antibody that blocks that. All right? In vitro, you can show that their antibodies actually block.
Why do they keep getting infected, though?
Well, all you need is one to get into the liver.
I see. Yes, of course.
That's it. And not only that, Plasmodium vivax, this is another species, of course, it actually sits in the liver for a long period of time without developing. Mm-hmm. Many of the sporozoites that get in don't even go on to develop right away. So there's a delay. Then you can get relapses and stuff like this.
By the way, are you interested where this work was done?
Yeah.
Würzburg, Germany, the Netherlands.
Wow.
Aachen, Germany, and Jena, Germany.
Nice.
At the Leibniz Institute for Natural Product Research. You know who Leibniz was?
Well, I know a little about Leibniz, but I don't know enough about him to say anything.
Mathematician.
Okay.
Didn't he invent calculus?
No, that was Pascal, I thought. Or maybe that was probability theory. Pascal was probability theory. Let's check it out. We got the internet right here. Why should we have to guess?
I don't have any internet.
I'm finished making an idiot of myself over the air because we have the internet. We can let the internet make an idiot of itself, and we don't have to take responsibility.
People love it when you make an idiot of yourself.
I know that, but I don't.
Gottfried Leibniz was a German philosopher and mathematician.
A philosopher.
He was. He has a prominent. He developed infinitesimal calculus independently of Isaac Newton.
Wow.
Okay, so he was a...
All right. So you know much more about that subject.
Well, I don't know much more. The only reason I know Leibniz is because I'm reading a book by Neil Stephenson.
Okay.
In which he fictionalizes.
Oh, okay. What did Pascal do? I thought he was probability theory, but let's just see for a moment.
Pascal?
Yes.
Pascal. You know, the listeners don't want to hear us search.
Sure they do.
No,
They don't. Come on, they're faster than we are. They've already got the answer. This is like Jeopardy, except the Jeopardy is that they won't listen.
Are you talking about Blaise Pascal, the French mathematician and philosopher?
I am.
There are a lot of Pascals in the world.
Another philosopher, mathematician, my goodness.
Made important contributions to the study of fluids and clarified concepts of pressure and vacuum by generalizing the work of Toricelli. You know,
Toricelli? He used to make Italian pastries and pasta.
No. He was
that was a Toricelli. That was another.
Okay, does that answer it?
No, it didn't say what kind of mathematics he specialized in.
Treatise on the arithmetical triangle, like this. Here,
really?
This is his triangle.
No, but where is probability theory? I thought he was interested in probability theory. Statistics. Statistics, that's what I thought. Okay, we're passing time here. Vince is looking over
I'm looking at the clock. 50 minutes. Do you want to do the other paper?
It's up to you. Or we could answer some emails and save that one for the next time. I'd have a much better reading on the deeper meaning of that if we could do that. But it's too bad because Trypanosoma cruzi attacks heart muscle, and this is Valentine's Day. But we can save it for another time. Post-Valentine.
Trypanosoma cruzi attacks heart muscle, and that's why we should talk about this on Valentine's Day.
We could talk about a different view of St. Valentine's Day massacre. In this case, it's like 12 million people with the infection.
Well, we do have some good email here.
Good.
So let's do them.
Up to you.
The first one is from, how would you pronounce this word, this name? Alaric. Aleric?
Aleric.
It's a nice name.
It is.
Alaric writes, hello, Vincent Dick. I just found your podcast, and I love it. You have some wonderfully witty banter.
Okay.
That's you.
Thanks, Aleric.
I'm only on my 4th episode. That's what you call a podcast segment, right?
Yes.
Yeah. And have a question pertaining to my original reason for searching out your podcast.
Uh-oh.
I'm working on the research portion for a short story I want to write about what the world would look like if we knew exactly how different parasites affect behavior. Think of it as a thought experiment dealing with the nature of environmental determinism and the concept of free will. Say, would people purposefully expose themselves to a parasite for a desired effect, or use one as an alibi for an act of bad faith? I may bring epigenetics into this story as well, but I'm not sure how it will fit. It is meant to be science fiction with a bioethical slant. Importantly, I don't want it to seem too sensationalist, though, so I'm trying to base as much of the story in reality as possible. Hence, listening to your podcast. Do you have any suggestions on where I should look for information about this? Do you have a podcast episode segment about this? Its validity as an idea, conspiracy theory, or reality? Also, I love the mini history lessons. Myself, I am a student of history of science, medicine, technology, and seriously appreciate it. A++, will listen again. Thanks.
Great. Okay. Well, remember we had an episode on toxoplasma, and when mice became infected, as all mammals are capable of becoming infected, mice seem to have lost their fear of being around cats. And that was the take on the behavioral change. And people thought, well, if that's possible, then maybe other parasites alter other behaviors too. But when you looked into it deeper and asked, why did the mouse lose fear of cats, the answer comes back, because it can't smell the cat. So the toxoplasma organism has knocked out the smell center, which allows the mouse to become on red alert before the cat gets too close.
Does it have a general effect on smell or just cat smell?
Well, on the mouse is ability to detect cat smell.
Just cat smell.
Yeah. So is that an altered behavior, or is that a loss? Behavior. It's a loss of a sense. It's a pathological effect of the organism on the sense of smell, which then results in a behavioral change or an apparent behavioral change. Because a lot of behavior is based on smell. In that organism, the mouse, its environment is odoriferous. And we know someone just downstairs from us, we mentioned his name before,
Richard Axel,
who got a Nobel Prize for showing the pathways between the nose and the brain and how smell is communicated, right? So apparently Toxo knocks that part out.
So it doesn't make the mouse more aggressive.
No.
Which is what people thought.
Yeah.
So that would be a perfect one for Aleric, right?
It would, but
if someone wanted to be more aggressive, they could take Toxo, but
not
it's
really.
Fiction. But it's science fiction.
No, but if you're a mouse, yes. But if you're a human being, what does that prevent you from doing that you could do before you lost your sense of smell? All right, that's
you
the
have
point.
to introduce some fiction into it, right?
You do have to do. So anything that humans react to with regards to smell, would be diminished in a
assuming the mouse extends to people.
Yeah. So let's take. You know what you could do here? Or you could create a wonderful story around a group of people who are highly selected by the food industry to act as tasters and smellers, and they're aroma experts. Mm-hmm. And you've got a competition between these two factions that hate each other because one always gets the jobs and the other one doesn't. So they learn about this parasite that can actually affect your ability to smell things. Yeah. And they sneak it into an hors d'oeuvre as they're having a big party. And the next thing you know, they get the jobs and this other group says, wonder why we have lost our sense of smell. It must have gone somewhere. And of course it went because the parasites took it away. That's the only way I can think of weaving this into some kind of a story that has plot to it, and then you could have murders, and you can go on and on like this. So,
all right. Now, the hookworms are immunosuppressive.
They are.
Could you weave that into some kind of plot?
A lot of them are immunosuppressive, sure, and a lot of them have cloaking devices. And you're asking me to make up stories. I think Aleric is going to be the one to do this. And
Just tell him the biology, and he'll make up the story.
Exactly right.
So we talked about toxo and mouse behavior in the toxo episode.
We did, and we had John Boothroyd here too, and he didn't think that it induced any other behavioral changes
in people.
Right? No,
Because there's some anecdotal evidence that it does something in people, but John said no.
It's like proving that the virus, the MDRX, causes chronic fatigue syndrome. It's anecdotal, and it's not.
When Rockefeller went to the South and saw people were lazy, that was because of hookworm?
No, because of the anemia induced by hookworm. The laziness, the inability to move at a required, I wouldn't call it speed, but
Got it.
They were just not industrious.
So that's another take or angle that Aleric could take.
Yeah.
Infect someone with hookworm to make them, if they're a runner or something, or competitor, make them lose.
Or you can alter human behavior that's related to some physical change. That's true, you could do that.
Well, we were just telling him the biology. So it's toxo and hookworm. Any others known to have an effect on behavior?
Well, you could have Giardia, and you can have rampant diarrheal disease, that'll change your behavior.
Yeah. If you wanted to knock someone out of a competition, you could give them Giardia, right?
You could do that.
And
it wouldn't be right.
They wouldn't be. Well, I think he's not. Doesn't worry about that.
No.
He's the author.
Yeah.
What else?
No, I'm not going to go down a big list on this one. Because
yes, you are. I'm holding a cup of coffee to your head.
You are good. I'll take it. I'd like it with sugar and milk.
Next one is from Robin. He tells us how to pronounce Shankar Ghosh. Actually, he sent us a little audio file.
Why would he do that, Vince?
What?
To pronounce Shankar Ghosh.
All right, so in the last episode, we discussed a paper from Shankar's lab. Shankar Ghosh is my chairman.
I knew you'd define it.
So I think you may have said Shankar.
I don't think I would have said that. Shankar.
Yeah,
Ravi Shankar. That's
Shankar. Right.
Yeah. So his first name is Shankar, but it's still the same pronunciation.
So he sent us an audio file of how to pronounce it.
Great.
Okay? I'll put
you.
that on the website.
Thank you.
He also said that Minhas in Portuguese means mines. Remember we were talking last time?
Yeah, right.
Minas Tirith, which is the famous Lord of the Rings castle, city up on the hill there. The city of the kings, right?
Right.
And there was on the paper, one of the places was Minas in Brazil, mines.
Right.
Okay?
Okay.
We ask, Robin answers.
Excellent.
James writes, "Hi, folks. You wanted to know how many New Zealand listeners you have, so now you know you have one more."
Excellent.
James is from Wellington, New Zealand.
Wellington?
Were you there?
No, that's in the North Island. Wellington's the capital of New Zealand.
Yes.
He knows that, but not a lot of other people would know it. But I was told while I was there that Wellington was built on a major fault line.
Mm-hmm.
Because I was in Christchurch, and Christchurch is in the South Island, and Christchurch also is built on a fault line.
Now we know we have 2 listeners in New Zealand.
No, we have 3. There's one in Otago.
One in Otago?
Yeah.
All right.
There may be more.
I've lost my place here, Dickson. Reading. Google Drive. The next one is from Peter. Jacob. I saw this in my inbox this morning and thought it would be a good TWiP pick of the week. This is quite nice, Dickson. Look. It's a photograph.
Oh, yeah.
Do you know what that is?
Well, it looks like flagellated protozoans.
Yeah, which one?
On the surface. Looks like maybe Trichomonas.
Yeah, it is.
I didn't
It's
read
from
it.
something called Cell Picture Show, which I'd never heard of.
Excellent.
Tricked Out Cows by Marlene Benchimol and Victor Dovalle. And Trichomonas fetus is a single-cell protozoan that infects the bovine reproductive tract and the intestinal tract of cats. Cows are infected by bulls that harbor the parasite on their foreskin. The resulting infection of cows can result in infertility, pyometra, and embryonic death and abortion. So this is a scanning EM of T. fetus, which has been colored green with a bovine culture of oviduct cells, pink,
Cool.
where adhesion of the parasites can be seen. Oh, they're beautiful. You know, I bet
They are beautiful.
part of the inflammation is due to viruses. Remember we talked about a paper where they showed in the vaginal mucosa, it's actually the the Trichomonas virus that is inducing an innate immune response, and that causes inflammation.
You did say that.
This is a beautiful picture.
Yeah. I think parasites are gorgeous. They are. They have beautiful symmetries and...
Next one is from... Sorry, I'm really
No.
sorry. Go ahead.
No, I was just going to say they're as beautiful as their hosts.
Why aren't your walls covered with parasite pictures?
They used to be.
I see.
They used to be.
Peter writes, Dear Vincent and Dickson. Oh, this is cool. You're going to like this. I thought this paper from PLOS ONE would be worth a mention on TWiP. Tapeworm eggs in a 270 million year old shark coprolite.
Ooh.
The fossil from Brazil has been found to contain the oldest known examples of tapeworm eggs. This specimen was so well preserved that a worm larva could be seen in one of the eggs.
Is that the Devonian era by any chance?
I'm going to look at the paper now. Do you know what coprolite is?
I do.
What is it?
It's a petrified piece of feces.
Yeah. This is from the Paleozoic. Do you know where that is?
Paleozoic.
Sorry, Middle-Late Permian.
Permian.
I'm sorry.
Okay, Permian, sorry.
Did you say Permian?
No, I said Devonian.
This is Permian. Well, what is the Permian? How long ago?
It's. I don't know the exact dates, but I know that the age of fishes preceded a lot of other things. So sharks arose during that time.
So that's an early...
It's early. It's early.
The Permian extend from 289 to 252 million years ago, the last period of the Paleozoic era.
Wow.
Anyway, these were found, these coprolites, fossilized poop, right, basically,
Yes.
bearing a cluster of 93 small oval elliptical smooth-shelled structures interpreted as eggs of a tapeworm. You want to see some pictures?
Sure. Vince has his iPad, and I'm going to bring mine in next time, too, because they're a lot of fun anyway.
So these are called cestoid eggs. The whole paper is one photograph.
Well,
actually, it's... Here, let's
it's
look
a note.
together here.
I wouldn't call it a...
Spiral heteropolar coprolite with cestoid eggs.
Yeah,
I don't know where this
the
is.
spiral valve of a intestinal tract of a shark is
Do
pretty...
you see cestoid eggs in there?
I can't see them yet.
No, yet. It's not going to happen.
It's over
What's
here.
This?
There they are. There they are.
Parasite eggs in a shark coprolite.
Very cool.
And then this one has a diagram which shows you.
Very.
And
very.
They see an embryo in there, right?
That's nice. You see the operculum? Little hatching area?
But this would be the embryo, right?
It's the embryo. That's right. It's the hexacanth embryo. See the little teeth on the...
What do you think about this?
I think it's very cool stuff.
Are you surprised that they're so old?
No. Of course not. I think parasitism arose the moment there were hosts.
Two parasites walk into a bar.
Go on.
Bartender says, "We don't serve your kind." The parasites say, "You're not a good host."
Right.
I'm here every week.
Try the veal.
So, as soon as life arose, there were parasites.
That's my contention. Or
Now,
very soon
what
thereafter.
good is this study, Dickson? Is this worth doing?
What do you mean, what good is it?
I'm asking you
it's
to
science.
explain to people what good is this.
It's science. It tells you, it shows you connectivity.
I always wanted to know what viruses were present a long time ago.
Vince, remember, what is the story about cestodes that's so intriguing?
You have to tell me. I don't remember.
All the parasites that we mentioned that were tapeworms
Yeah.
had intermediate hosts,
Okay.
right? And this would be no exception.
So these are...
So there must have been...
we don't know what kind of cestoid this is, obviously.
No, we don't. But judging by the shape of the egg and everything, it's got a little operculum. It looks a little bit like a Diphyllobothrium. Now, it won't be a Diphyllobothrium. It'll be some related species to that, right?
Yeah.
And that goes back a long ways. But remember, Diphyllobothrium employs little copepods, right? An intermediate fish, and then the big fish. Okay, that's its life cycle. So this means that during this time period, even so long ago, there were complex life cycles in the world. And that's the speculation that this elicits, from me at least. That's intriguing.
Do you think they could get some nucleotide sequence out of these eggs? Would that be interesting?
It would be fascinating.
This is the earliest fossil record of tapeworm parasitism of vertebrates and establishes a timeline for the evolution of cestodes.
See that? Wouldn't
you like to be doing this stuff still, Dickson?
We are doing that right now.
Oh, you're right.
Secondhand. We're doing it. We're sort of like we're looking through the glass mirror and seeing what our lives could have been like if we had continued.
Listen, I don't mind doing this at all.
No, it's fun.
And that's it for our email is dwindling because we have no listeners left.
Really? I mean, we have no more on this list?
That's it.
Really? We were caught up to date?
Yep.
Wow.
No more.
So if you're listening out there, start writing again. We're lonely.
Well, in fact, Dickson, the more you podcast, the more email you get.
Yeah, I've been told that, actually.
And we had a long gap of a month. So
We did. People lost interest. They just lost
Well, they don't lose interest, but they went away because we went away.
I guess.
Now, if we come back, they may come back. But if they don't, my feeling is if we don't get emails we do the next 4 or 5 TWiPs.
You just trash it.
We're going to just can it.
Yeah,
which is how we started this show. You see, when I say that, you should say, "No, Vince, we have to keep doing it." But you say, "Sure," and that's anyway. TWiP can be found at microbeworld.org/TWiP and also on iTunes. That's where all the shows are kept. And if you want to help us. You probably don't because you're not there anymore anyway. So
Please.
Go over and leave a comment or a rating.
This is Pledge Week on This Week in Parasitism.
Send your emails. If you do want to send emails, twip@twiv.tv.
Right.
Now, the reason I ask you to send them to twip@twiv.tv is so that I can sort them and know which ones are for TWiP and which ones are for TWiV and which are for TWIM. Now, I know I should have twip@twip.tv, but TWiP.
Vince, isn't it interesting to notice that TWiV, TWIM, and TWiB all have cross-reactivity?
Where'd you get TWiB from?
I just made it up. So you have TWiP. There are parasites that have bacterial infections, like Onchocerca.
Right.
And there are parasites that have viral infections, like Leishmania and Trichomonas. So you've got cross-connectivity between TWiV, TWiP, and TWIM.
Of course.
So why we should be more gregarious?
In what sense?
Perhaps we should do a mix and match show one day.
Well, I think people listen to what they're interested in. But I also think that
we've never done a virus story on a protozoan on TWiV.
Sure we have.
No, we haven't. Not on TWiV.
Oh, on TWiV? Yeah, we did.
No, we haven't.
You don't think so?
I don't think so.
All right.
You've done 250 some odd shows on that.
220.
And not one showcases the virus infections of protozoan parasites. Not one. But you should.
I will have to look to make sure you're right. You might be right.
I don't remember.
On TWiP, we did a virus of parasites on TWiP.
The TWiP we did.
I mean, I thought I was saving it for TWiP in that sense to be good.
No, I think your audience is huge.
Much bigger on TWiV, I know.
They're enormous.
Why is that?
Because you do a better job than I do.
No, not at all. First of all, is the frequency. It's regular every week.
There are more viruses.
The other is that I think people are fascinated by viruses and less so by parasites.
And I don't agree with that at all. I think once they heard about the parasites, they're fascinated. But I think there are more virologists out there, frankly. Virology is a very, very big field because it causes a lot of diseases that are really serious.
Dickson, I will argue that parasites cause a lot of disease, too.
Well, viruses are parasites.
I read today that there are 216 malaria infections at this moment.
Billion. That's 2.7 billion. That's right.
So, I don't think...
but they don't get the play that some of these virus
You know what Tony
infections...
Fauci said? I said to him, what's the most important infectious disease? He said AIDS. I said, what's next? Do you know what he said?
Malaria.
Malaria and TB.
That's right. Yeah, because that's a mantra. It's a mantra.
Really?
Yeah. And you remember what we said about that too, right? On one of our TWiP shows, we proved that schistosomiasis, hookworm, trichuris, and ascaris cause more human suffering than these three combined will ever cause.
Yeah, that's right. I remember that.
And yet these things receive short shrift, and these get all the press.
Dickson, next time, let's do another one in 2 weeks. Okay. I'm going to go back and comb our old emails for suggestions. A lot of people have asked us to do things, we haven't done them. So
sure.
Let's do that.
Fine.
I feel bad because before we stop, we should satisfy everyone's requests.
I agree.
You're not going to do it. You're not going to beg, are you?
No. I don't beg.
Dixon Despommier is at trichinella.org, verticalfarm.com, and medicalecology.com. Hey, how is the vertical farm?
It's doing great, Vince. It's doing
So
great.
you're wealthy.
No, your book. You're asking about the book or the concept?
No, the concept.
It's doing fantastic.
Yeah?
I don't make a penny on that stuff.
That's
I
a
just want to
shame.
see it happen. No, it's not a shame. That's not how I want to make a living. I think I just want to see it done.
You want to eat lettuce from a vertical farm.
I've already done that.
And you recently visited one in Sweden, right?
Well, they didn't actually have a ongoing construction at that point. They were still waiting for their building permits from the city.
What's your next trip? Germany?
No, Chicago.
You're going to see vertical farm there, right?
There are 2 there now.
That's cool.
Yeah. And actually, there's one inside the Museum of Science and Industry as well. But
I thought you were going to Germany.
I am.
When?
March 20th.
And you're seeing a farm there?
No, I'm going to consult with the government of Germany, actually, that wants to get involved in this. So it's part of a larger
Are you
meeting.
going to Berlin?
Yeah, I am. Yeah.
Okay.
It's going to be fun.
I'm Vincent Racaniello, and you can find me at virology.ws. The music that we use in the beginning and ending of TWiP is composed and performed by Ronald Jenkins. You can find him at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.